Hi everyone, we will continue to talk about the citric acid cycle. So before we start, we want to recap the TCA cycle reaction we have introduced last time. As we talked about, the citric acid cycle produces high transfer potential electrons, ATP, and CO2. So then the next reaction of the citric acid cycle is shown here. This is how it occurs, actually. So first, the condensation of an acetyl unit from acetyl-CoA with oxaloacetate. The isocitrate dehydrogenases and alpha-ketoglutarate dehydrogenases, thus both enzymes catalyze successive decarboxylation so that two carbon atoms leave the cycle in the form of CO2. And then step 2, so 4 pairs of hydrogen atoms leave the cycle in 4 oxidation reactions. 2 NAD+ molecules are reduced in the oxidative decarboxylation of isocitrate and alpha-ketoglutarate. 1 FAD molecule is reduced in the oxidation of succinate, and 1 NAD+ molecule is reduced in the oxidation of malate. So, in addition, 1 NAD+ molecule is reduced in the oxidative decarboxylation of pyruvate to form acetyl-CoA. One compound with high phosphorus transfer potential, usually ATP, is generated from the cleavage of the thioester linkage in succinate CoA. So, 2 water molecules are consumed, one in the synthesis of citrate by the hydrolysis of citric-CoA, and the other in the hydration of fumarate. So let's summarize this reaction. And you will see in this table is present all the details about this reaction, and it's kind of complicated. So then we'll talk about the regulation of entry into the TCA cycle and metabolism. Entry into the TCA cycle and the rate of the cycle itself are controlled at several stages. So the first one, the synthesis of acetyl-CoA by the pyruvate dehydrogenases complex is a key irreversible step in the metabolism of glucose. The oxidative decarbonization of pyruvate to acetyl-CoA commits the carbon atoms of glucose to oxidation to CO2 by the citric acid cycle or incorporation into lipid. High concentrations of NADH and acetyl-CoA inform the enzyme that energy needs of the cell has been met. Then step 2. Phosphorylation of the pyruvate dehydrogenases component E1 by pyruvate dehydrogenase kinase PDK switch off the activity of the complex. So specific kinase phosphorate and inactivities pyruvate dehydrogenase DPDH and phosphatase activity the dehydrogenases by removing the phosphyl group. The kinase and the phosphatase also are highly regulated enzymes. Then it goes to the third step. So the pyruvate dehydrogenase complex is regulated to respond to the energy charge of the cell. And it shows here that like it shows that the complex is inhibited by its immediate products and NADH and acetyl-CoA, as well as by the ultimate product of cellular respiration, ATP. And it shows in B that the complex is activated by pyruvate and ADP, which inhibit the kinase that phosphorylates PDH. So how to control the TCA cycle? The citric acid cycle itself is controlled at several points. It is regulated primarily by the concentration of ATP and NADH. The key control points are the enzymes. So first is called like isocitrate dehydrogenase, which is stimulated by the levels of ADP and inhibited by ATP, alpha-ketone glutenate dehydrogenase. And the second one is alpha-ketoglutinate dehydrogenase. And this enzyme is inhibited by succinyl-CoA and NADH and a high energy charge. Thus, the rate of the cycle is reduced when the cell has a high level of ATP. TCA cycle as a source of biosynthesis precursors. So we'll talk about this. And as a major metabolite hub of the cell, TCA cycle is an important source of intermediates for biosynthesis. For example, many of the carbon items in phosphorin come from succinyl-CoA, and most of the amino acids are derived from alpha-ketoglutarate and oxalic acetate. So it shows here the red arrows shows like intermediates for biosynthesis when the energy needs of the cell are met. Intermediates are replenished by the formation of oxalic acetate from pyruvate. This biosynthesis process will be considered in subsequent chapters. So then let's talk about glycoxylate cycle. So although oxalic acetate is formed in the TCA cycle, the 2-decarboxylation that takes place before the regeneration of oxalic acid prevents the net conversion of acetyl-CoA to glucose. Glycoxylate cycle is a metabolic pathway that allows the conversion of acetyl-CoA generated from fats stored into glucose in plants and some microorganisms. The overall reaction is as follows. In plants, this occurs in organelles called glycosystems and is especially predominant in oil-rich seeds. For example, sunflowers, cucumbers, and kind of beans also. So let's talk about the differences between TCA and the glycoxylate cycle. This cycle is similar to the TCA cycle, but bypasses the 2-decarboxylation steps of the TCA cycle. Another significant difference is that 2 molecules of acetyl-CoA enter per turn of the glycoxylate cycle compared with 1 in the TCA cycle. Okay, so that's all for the citric acid cycle, and thank you for your attention. Bye.